Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this is number 40, going out on December the 19th. And it's another three-person podcast, although two are in the same conversation. This will be the last podcast before the holiday season, considering most people switch off at least a day before Christmas and aren't back online until after New Year's Day. So we'll be back then. But before that, we have a great episode for you. Last week we had conversations about an artificial intelligence event in Sweden with Jakob Andersson, as well as about genetic medicine for eye conditions with Ankyra Therapeutics CEO Rodolphe Cleverel. We also chatted with Mobile Ion Systems CEO Melissa Sherman. This week we're talking about skin and wounds with Ned Swanson, President and Chief Medical Officer at PolarityBio, and Nikolai Sopko, Chief Operating Officer, Chief Scientific Officer, and Director at PolarityBio. And we are discussing anchored immunotherapy with Howard Kaufman, CEO of Ankyra Therapeutics. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us or sponsoring a podcast episode, or indeed inviting us to an event somewhere to cover. First, let's take a look at the news you may have missed over at deeptechdigest.com, and yet again, in spite of the fact that it's the holiday season, there was a lot to squeeze in. Ankyra Therapeutics raised $100 million for its global transformative therapies. Vivacta Bio showed promising results for its in vivo CAR-T therapy in non-Hodgkin's lymphoma, and PanCancer T raised €10 million for its first in-human trial of a novel TCR T-cell therapy for triple-negative breast cancer. A rapid diagnostic test is being hailed as a breakthrough in the fight against Klebsiella. Cythera has been unveiled with $47.5 million in funding to advance oral medicines. And Eli Lilly is building a $6 billion facility in Alabama. A breast cancer vaccine saw its trial endpoints met with a 74% immune response. The FDA has approved a generalized myasthenia gravis drug, and the first precision therapy prostate cancer drug has been approved. InnoCare is moving on to a Phase 3 trial after achieving its endpoints with a treatment for systemic lupus erythematosus. An immune switch is a potential target to curb chronic inflammation in cirrhosis, and Sobi is taking on gout after acquiring Arthrosi Therapeutics. And you can read all of these and plenty more at deeptechdigest.com. Okay, let's get to the interviews then. First, this week, most people know what immunotherapy is. But what about anchored immunotherapy? Well, to tell us about it and how it's being used in the fight against cancers is Howard Kaufman, CEO of Ankyra Therapeutics. First, then, I wonder if you could tell me a little bit about the company.
Sure. So Ankyra Therapeutics is a small biotech company, and Ankyra is the Greek word for anchor. And so we are developing what we term anchored immunotherapy. The problem with a lot of drugs, not just immunotherapy, is that you need to get high doses of the drug to treat a cancer. And yet, when you give more of the drug, you get toxicity, especially when you're giving these things intravenously. So the concept here is that we can actually anchor the drug at the tumor site, which is where it needs to work. And we do that by physically linking a drug to aluminum hydroxide, which then acts as the anchor. And what we found is that if we inject that into a tumor, it can hold the drug there, allows us to give very high doses of the drug where it's needed most, which is where a cancer is growing, and it's not going anywhere else, so there's less toxicity. And in early studies in mice, we found that often one dose would be curative in fairly well-established tumors, which is why I came to the company, because it's very unusual to see that kind of potency in mouse studies. So the first target that we went after was an agent called interleukin 12. And interleukin 12, which we sometimes shorten as IL-12, is a cytokine. So it's a natural body, in a sense, immune hormone that helps to activate T cells, which we think are the cells that can actually recognize and kill tumor cells. And so actually, when I was a fellow many years ago, interleukin 12 was given to patients and it actually seemed to work. But as the dose went higher and higher, there started to be some really critical side effects. People got liver toxicities, their white counts went down, and then when the studies got expanded, several patients started dying from it. And so it never got continued because it was felt that in order to get enough dose in to help the cancer, you were going to get into too much toxicity. And yet other cytokines like interleukin 2 and interferon went on to get approval to treat cancer, but you needed very high doses. And actually, we hardly ever use those drugs today, not because they don't work, but because they actually are so toxic. So we felt if we could get interleukin 12 to work through this platform, we might really unlock the potential of that to treat the cancers without the toxicity. And frankly, that's exactly what we've been finding. We've started a phase 1 study. The study is going on currently in the United States and Canada, and we've really enrolled patients who have a whole range of solid tumors. So we've had patients with melanoma, head and neck cancer, skin cancers of other types, a number of patients who've had GI cancers. We had 2 breast cancer patients, 2 bladder cancer patients. And overall, we're seeing about 60% of the patients treated have achieved at least stable disease, or what we call disease control, for at least 6 months. And several patients have had what we would call an objective response, which is sort of rare to see in a very first-in-human phase 1 study with just a single agent like this. These are all patients who've already failed standard of care treatments. So we're obviously quite excited about this, and we're currently developing a broader pipeline. So after the cytokine, we've recently been able to put on a monoclonal antibody onto the anchored platform. And monoclonal antibodies are widely used throughout human medicine to treat a variety of diseases, including cancer, autoimmune disease, a number of GI diseases. And antibodies are sort of, in a sense, similar to cytokines, that high doses are often very helpful for either ameliorating the disease or treating the disease, but they can have a lot of side effects as well. And so the fact that we can now successfully put a human monoclonal antibody into the anchored platform really opens a door to us to treat a wide variety of diseases. So that's sort of a little bit about what we're doing and where we're at.
All right, perfect. You mentioned how you had come to be involved with the company. Could you go into a bit more detail about that? Because your background is quite interesting.
Yeah, thank you. So I really was in academic medicine for a long time, and I was interested in oncolytic viruses. I had done my fellowship work developing one of the first recombinant viral vaccines to treat cancer, and then I got very interested in oncolytic viruses and was part of the original, what we call TVEC study, which is one of the viruses that got approved really throughout the world for treating melanoma. And that got approval back in 2015. And so I was very interested in local therapies. And I guess now about 10 years ago, I had the opportunity to go back into industry at an oncolytic virus company, which I did. And I was not really looking for a job when I got a call from the chief scientific officer at Ankyra, and he said, Howard, you have to come here. We need a chief medical officer, and the science is really spectacular, and it's just such a good fit for your background. So I came and I looked at the data, and it was pretty remarkable. That's when they had the mouse data, again showing a single treatment would cure these mice who had really large tumors. And I wasn't even going to come. And then I talked to the founder of the company, and it's a company that spun out of MIT. And by the end of that phone call, I felt like I had to come here. And it was just too exciting of a finding to kind of pass up. And actually, I brought some of my team with me from the other company. And about 6 months into moving over here, they asked me if I would be interested in the CEO position. So I felt that was a unique opportunity, and I accepted that, and I've been doing it now in March, it's going to be about 5 years.
Are other companies doing things with anchored immunotherapy as well, or is it just your company?
It's getting some attention. So I would say that the idea of somehow making these drugs safer so that you can get higher doses to patients is not exactly a new concept. But I think the way we're doing it, we're really able to achieve much higher doses of the drug. So some people are directly injecting, for example, RNA into a tumor, and there's a number of different companies doing that, and that does seem to be offering some activity. The challenge there is that once the RNA is transcribed into protein, those proteins basically can leak out of the tumor, and then they become systemic and they can cause side effects. So some of these are really hitting a cap where they can't put the doses any higher. Other companies are trying to do what's kind of called protein masking or capping, where you take an active protein like an IL-12 and you put some sort of cap on it so that when it's circulating in the system, it doesn't do anything. But then when it gets to the tumor site, an enzyme can uncap it or unmask it, and you can get the activity from it. And that approach is actually showing some promise as well. But of course, the enzymes have to be working at the tumor site, which is not always happening, or it's not always known if they're going to be there. And again, once you uncap it, the drug is then free to float away from the tumor. So I think we have a really interesting way to kind of keep the drug there. And it's really allowed us to give almost 1,000 micrograms more drug than these other groups have been able to give. So we're definitely seeing pretty high doses. And to date, we haven't seen any what we would call dose-limiting toxicities in the patients. So these would be, really severe side effects where we may not be able to give higher doses. So obviously we have to study this in more patients and specific diseases to really understand how well it works. But so far, in the early phases, you really want to make sure that your drug is safe. And in that regard, I think the data that we have is looking very promising.
Are there any specific challenges associated with anchored immunotherapy?
It's a good question. One of the important things is, unlike oncolytic viruses that have a biosafety concern, you have a live virus. So that's obviously not something that's easy to send out to, small clinics, and just transporting a virus around the planet is not an easy thing. So we don't have any of those challenges. We've actually had very good stability with the product. So once we make it, it's looking like the protein itself is stable at refrigerated temperatures for 4 years or more. We're doing ongoing studies. So that makes it really easy to ship it around and, to send it to smaller clinics. I think our biggest challenge with this is it is a local therapy. So it's something that we directly inject into a tumor. So in the case of skin cancers, they're often on the skin. So it's like pretty easy to do. In the case of patients who may have tumors that are in other areas of the body, it can be a little more challenging. But we use interventional radiology or ultrasound, and there's very few places that we really can't get to where we can inject the tumors in. And because it looks like they stay in the tumor for a long time after the injection, we don't have to do this all that often. Right now, we're doing it every 3 weeks, and we're thinking about going to every 6 weeks. And, that's something that we need to work out in the early part of development. interestingly, we couldn't do much of the work in the mice because they all got cured of their cancer after a single dose. So it was very hard to give them multiple doses.
Is it something that you anticipate would be used as a standalone therapy or in conjunction with other drugs?
Yeah. So again, we're at the early stages of the development. I think there may be room for both. I'm quite excited about using this as what would be called a monotherapy or a single agent in what we call the neoadjuvant setting. So these are patients who have not had surgery yet. And recently, there's been a lot of data that for many immunotherapy agents, giving them before surgery is much more effective than giving them after surgery. So I started as a melanoma doctor, and in melanoma, there's actually been a large randomized study where patients got immunotherapy either before surgery or they got it after surgery in what's called the adjuvant setting. And then they followed patients to look for long-term survival. And the patients who got smaller doses of the drug in what we call the neoadjuvant before surgery setting actually had much better survival than patients who got adjuvant. And I think our agent is especially well suited to that because we might be able to get away with like just one or at most two doses prior to a surgery. You do the surgery, and what the drug would do, it really allows you to use that cancer that's in the patient to educate the immune system to what they need to destroy. So when you get a vaccine, what you're basically doing against, say, an infectious disease is you're giving the immune system part of the pathogen so that it kind of is educated as to what it needs to look out for. And then it can go, and if a person gets infected with that pathogen, the immune system will respond to it. Well, similar thing has to happen for cancer. We've got to teach the immune system what it needs to go after. And so using the cancer that's already there is a really clever way to do it. I think when patients have more advanced disease and it's spread, it's a lot harder to treat. And this is where I like our drug in combination with other drugs. So we have some what we call biomarker data where we've actually collected biopsies from patients treated with our drug. And what we're seeing is actually a very significant increase in a protein called PD-L1. And that is very well known to many oncologists because that is the target for immune checkpoint blockade drugs. And we know that those drugs work better when PD-L1 expression is high. So we've actually enrolled a few patients in our study that didn't respond to those drugs. In part, we think because their PD-L1 was quite low. And after our drug, they had a very significant upregulation of the PD-L1. And some of those patients actually went on and were able to get checkpoint blockade after coming off the study and have actually experienced pretty good long-term survival at this point. So we've recently, and I mean within the last 4 weeks, we've actually started a study in lung cancer where we're actually combining our drug with anti-PD-1 agents, which are now approved for treating non-small cell lung cancer.
Could you tell me a little bit about your clinical trials so far in the Lantern trial?
Yeah, so we are in the process of completing our phase 1. It's nearly over. We're like one patient away. And then we are doing what we call an expansion cohort where we're combining our drug with another agent called cemiplimab. It's another anti-PD-1 in patients who have squamous cell skin cancer. And we're quite excited about that. And we've just started enrolling patients to that study. And then we have our separate study in lung cancer that just started. And we are looking at patients in what we call first line. So this will be the first therapy they got where we're going to give them the combination. And then we're also treating patients who've already failed first line. And we're giving them our drug in second line with the anti-PD-1. And our hope is that we'll be able to get a better idea of which patients are most likely to respond from the drug. And then obviously we would want to expand that up in the future.
You recently gave a presentation at SITC, and you've also been busy with a Nature communication piece as well. What's the reaction been like to both of those?
I think it's been good. My academic colleagues are quite excited about this. So we've actually in some ways had more interest than we have resources to accommodate. So it's good to see that the people that are using the drug are excited about it. One of the things we reported at the SITC, the Society for Immunotherapy of Cancer meeting, is in the patients that were treated in the early part of the phase 1, we were looking again for any biomarkers that might predict response. And we saw an interesting finding that at baseline, this is when patients signed up to be in the study, we measured the number of a cell type in the blood called NKT cells. It's a very small population of the T cells that exist, and they have an appearance of both a T cell and an NK cell. These are two types of blood cells that are known to fight infectious diseases and cancers, and a very, very small percentage have what looks like both NK and T cells, so they're called NKT cells. And some of the patients had sort of high levels of those, and some patients had low levels. And it looks like the patients who had high levels all seem to get a benefit from our drug. And what's interesting about that is these cells are known to have very high numbers of interleukin 12 receptors on their surface. And so it may be that our drug is really able to activate these cells, and they may be playing an important role in fighting the cancer for these patients. If this is true, it could be an important way to better select patients in the future who might benefit from the drug, and also avoid giving the drug to patients who may not benefit. So we obviously need to increase the numbers. This is, based on about 40 some patients, so it's still a very small sample size.
And is the ultimate goal with this kind of treatment to completely reverse the cancer and get rid of the tumor completely, or to just stop it in its tracks so that it becomes a manageable disease?
Yeah, another good question. Obviously, we'd like to get rid of it completely. And, one of the nice things about immunotherapy agents is they often can do that if you can really engage the immune system. And a lot of times people don't realize this, but immunotherapy, it's not really targeting the cancer, it's targeting the immune system and really trying to get that immune system to get active. And when that happens, we think we actually can get patients cured of these tumors. And even if we can't, if we can control it, turn it into a chronic disease, that's not necessarily a bad outcome. So we would certainly take that. But we're really quite hopeful that this will behave like a true immunotherapy drug and potentially can be curative at the end of the day. Whether these things can have, preventive potential, we don't know. Typically the way we study drugs in oncology is we take patients who are dying of metastatic disease and we study it in those patients first. And if it looks good there, which is a really hard thing to ask of any drug, then we study it in patients who have a little bit less active disease, usually in the adjuvant setting. And only then, if it looks good, do we go into early phase and talk about prevention. But, I'm already excited about the potential of these agents in those settings. And, we can identify patients who have what we would call kind of high risk cancers that we know are at risk for coming back or spreading. And can we get to them earlier with a drug like this? And again, I just can't stress enough that while we're seeing some side effects, they're very mild. We're seeing things like small fever of a temperature of about 99, 100, and it goes away with a Tylenol. We're seeing some chills and feeling tired, and it's lasting for a very short period of time. And so far, that's mostly what we've seen with this. So we're not seeing some of the more severe or permanent side effects that you can see with, say chemotherapy or systemic, immunotherapy agents.
Does the stage at which or a diagnosis is made make a difference to the efficacy?
We think so. That's what I was sort of talking about in the neoadjuvant setting. So I think if we can get there earlier, we think it will be a lot better to treat them. It's been challenging to do those studies because usually we have to go to the patients who have, more late stage disease, say stage 4, which is what we've been looking at so far. But the FDA has already granted us kind of an initial okay to look at earlier stage patients. We're quite excited about that. Like many biotech companies looking for more funding to support that study, but we're hoping we're going to get there. And whether this will work better in earlier stage remains to be seen, but we think that may very well be the case.
And speaking of funding, could you tell me a little bit about some of the partnerships and funding that you're looking for?
Yeah, so we're currently supported by a very well-known group of private investors. We've also had some very good relationships with big pharma companies. So we're currently in a very strong relationship with Johnson and Johnson. And I'm really excited about that relationship because they have a group internally called their Interventional Oncology Group that's really interested in bringing these what we call local therapies to patients. And they're especially interested in really doing something about lung cancer. And so we've been very aligned on the mission, and it's been really nice to work with them. And it's been a very good partnership. So they brought a lot of resources to the table, and they've really given us a lot of support to push our studies to the point that we're at. We also have a very strong relationship with Regeneron, who are supplying the cemiplimab for our skin cancer study. And of course, they're one of the leaders in treating squamous cell carcinoma, which is actually very common. It's the second most common cancer worldwide. And actually, more patients are now dying of squamous cell cancer than are dying of melanoma. And cemiplimab has really been a very important advance in the treatment of that disease, but it doesn't treat everybody. So we're quite excited about combining our drug with their drug. And, in the near future, we're hoping to look for additional investors as we want to expand our studies now that we're seeing some exciting things and we have new products to bring to the table.
You were mentioning the difference earlier between the people that respond positively to what you're doing and those that don't. Are you also studying, or will you be studying people that don't respond well so that you can get some kind of idea as to why that is and maybe circumvent that in the future?
Yes, definitely. So what you're bringing up is, all drugs we have for disease, patients can develop resistance to them. They may not respond up front, or they may respond for a while and then not respond later. And this is a really important thing to understand. So from the get-go, in our phase 1 study, we have been collecting blood and we're getting biopsies on patients before and after treatment to study those exact things. And we are actually finding things already. So the amount of data that's come back is actually kind of enormous. So this is where we're now starting to use things like artificial intelligence to help us with the sheer volume of data. And so one of the early things that we've seen is locally, when you look at the tumor site, following treatment with the drug in patients that are responding, there's a very high upregulation of all sorts of genes related to activating the immune system, exactly what we would expect. In the patients not responding, they're not doing that. So those genes are simply just not getting activated for some reason. And now we have to dig a little deeper to understand what is blocking the ability of those patients to generate that response. So it could be that there's something in their immune system that's just not really able to respond, and we may need to give them some additional help to get that response. Or it's possible these cancer cells can be very clever, that they're producing something that is kind of getting in our way. And if we can understand what that might be, then yes, we may be able to come up with a more logical combination that could really help those patients.
On top of all the things that you're already doing, are there other things in the pipeline?
Yes, we actually just announced at the recent SITC meeting that we are now generating a CD40 monoclonal antibody agent. So CD40 is an interesting target. It is a receptor that is found on myeloid cells. So again, these are part of the white blood cells that really help to start the whole immune process and usually activate the T cells, which are the cells that go and kill the tumors. And early on, about maybe 10 years ago, people studied CD40 antibodies in patients. And a group at the University of Pennsylvania were the first to report patients with pancreatic cancer seemed to respond a bit to this drug. However, like a lot of these immunotherapy agents, once they gave a higher dose of it, they started seeing a lot of side effects. So patients got liver toxicity again. They got like blood clots and problems in the clotting system. And so they just weren't able to get a high enough dose in. And so we think the anchored platform would allow us to get the high dose in without the side effects. And so we're really thinking that that will be our next product. We're working hard to now start the manufacturing for that agent. And if all goes well, we're hoping that we could be able to get this into patients hopefully within the next year or so.
And where do you see anchored immunotherapy going, or how do you see that evolving?
Again, if everything holds up with how we think this works, it means we really can take effective drugs, get very high doses into patients that need those high doses, and do it in a very safe manner that really is not impacting patients or their quality of life in a significant way. So, we would like to work with big pharma companies who have drugs that, they may be on the fence with because of side effect issues, but they think have been effective. And while I think there's a whole range of cancers that we can think about going after, I think this technology could go beyond cancer. I think we could use this, for example, to treat autoimmune diseases. These patients often, the trick there is you're turning the immune system off, but you're still targeting the immune system. And a big problem with these patients is if they're on systemic immunosuppression for a long time, they can get cancer and they get infections because their immune system is being shut down. We might have a way to shut it down locally. So, for example, patients who have bad arthritis and you have one joint that's a problem, this is really well suited to get rid of the arthritis in that joint and not bother the rest of the body where, the side effects may occur. It also could have implications for transplant patients who also need their immune system suppressed to keep an organ going. And we're really quite excited about studying our drug in the transplant population because right now they can't get immunotherapy because they run the risk of rejecting their organ. We think our drug is going to be so safe that likely would not happen, that the immune system is only going on locally where the disease is. And so this is where I think we could take this. So I think it could have very broad implications for many different human diseases. And in fact, I might add, beyond humans, early on, we had a canine version of our drug. And so we actually had the opportunity to do a phase 1 study in dogs. And so we actually tested a canine interleukin 12 in dogs with melanoma. And it was a study done at the University of Illinois Vet School with a veterinary oncologist we worked with. And there we tested the drug in 18 dogs who had advanced melanoma, and about half of those dogs achieved good disease control, and some of them have been disease-free for several years. And so that was so unexpected. We actually out-licensed it to a company called Jenga Biosciences, which is focused on animal cancer care, and they're now developing this for 4 different cancers in dogs, which is really quite exciting. And so we think the whole anchored technology and the platform could have many implications, beyond just treating skin cancer.
Is there anything that you wanted to cover that we didn't talk about?
I'm really grateful to the team I have that's really dedicated to this and the patients and the families who agree to go into these studies. We learn a lot, and I still see patients, so it's very close to my heart. But I think a lot of the drugs that we're developing these days have so much more potential. The odds of something working are much higher today than they've ever been. And this is a time we have to keep supporting science and drug development because we're really making a difference. It's really just so satisfying for me to be able to see this go from, an idea just 2 years ago to we're now in patients and dogs and seeing some of them doing really well. As I was saying, I think drug development is a difficult process. And again, I've had a really good team here. They've been very loyal and worked hard. And we do have a system down now where, once we identify a drug that we think is important, we can get it anchored onto this platform almost within like a 3-month period. And we've developed the assays to test it very quickly. So recently, once we got the green light to do lung cancer work, we basically went from starting it up, not even having a protocol, not having any sites, to getting the IND approved, getting the first patient in less than 6 months, which is almost unheard of. We put a number of systems in place. We use the artificial intelligence where we need it. And, we focus on what absolutely needs to be done, to get this into the patients.
So now you know what anchored immunotherapy is, if you didn't already. One more string in the bow against cancers, and I guess we need all the help we can get. Now we're heading to the skin, and one interesting quote that came from the company PolarityBio, and I'm paraphrasing just a little, is that the skin could be the first organ to fulfill the promise of regenerative medicine. To explain, we had a conversation with two people from the company, Ned Swanson, President and Chief Medical Officer, and Nikolai Sopko, Chief Operating Officer, Chief Scientific Officer, and Director. Maybe then first we could start with a little bit of background on the company.
Yeah, I'm Ned Swanson, the Chief Medical Officer of the company. Nick's the Chief Operating Officer and Chief Scientific Officer and a board member. But to dive into PolarityBio, it's a clinical stage biotech company developing autologous skin multicellular therapies to address unmet needs in wound healing. So our first product, Skin TE, has an open IND with the FDA. We just finished our phase 3 pivotal trial very recently, and we'll be applying for a biologics license application for that product. But as a high-level overview of Skin TE, it starts with a piece of the patient's own skin. So the provider takes a full thickness piece of skin from the patient, very small, 3 centimeters squared, basically in the shape of a larger almond, if you will. So a football-shaped ellipse. They sew that up in a straight line. So the patient's not left with a wound. It's just it'll heal as a linear scar. They send that to our company in Salt Lake City, where we turn it into Skin TE using all of the skin. So all the cells and the tissue matrix that's sent to us, we use to make Skin TE. It goes back and they use it within 10 days of taking the skin. And it comes in a syringe, comes out almost like a sticky oatmeal. And they apply that in the wound. It engrafts into the wound bed. And then it's able to proliferate and differentiate the skin cells across the wound bed, as well as signal to the surrounding wound bed to heal. And I can let Nick go into, exactly what Skin TE is on a more detailed level, if that makes sense.
Yeah, so as Ned mentioned, we get a healthy full piece of skin from the harvest using this is the little stencil that we provide the providers to trace out on the patient. So quite small, a little bit larger than an almond. And the key there is that it's autologous. It's using the patient's own cells. And the reason for that is, as we're developing the product, looking at the need for wound healing, by definition, a wound is a lack of cells. And so when we look at a lot of approaches to wound healing, even going back thousands of years with just wet gauze, all of those approaches are generally providing a temporary cover, hoping that the body's intact cells will be able to marginalize and move in and close that wound. But, we can all recognize that the skin has incredible regenerative capability. It's the third most regenerative organ in our body. It's replacing 10 million cells a day, which means we're shedding 30 to 40,000 cells a minute. And so there's already this tremendous regenerative capacity of skin, but we know that there are wounds that are too large, or in the case of chronic wounds, have been around too long that Mother Nature has just not evolved to deal with. And so our thought process was, how can we take what Mother Nature already does so well and optimize it for these wounds that she honestly has never really encountered before? And so that's where one of the first aha moments was, we have to get some of the skin from the patients so that we can get cells into the wound and they're not rejecting. The next biggest aha moment was understanding, being in Ned and I in the wound care space, and in regenerative medicine really in almost 20 years, is that a lot of the early approaches to regenerative medicine was, okay, what's sort of the key cell? What is this holy grail cell in whatever organ that we're looking at that we can isolate, maybe expand, and get that to do everything? But when you look at most organs, and especially skin, there's 50 different cell types in skin. And understanding that there's really an orchestration of multiple cell types in order to really enact wound healing. So how can we leverage all of those cell types and not just our one favorite cell, not just the keratinocyte, not just the fibroblast or your LGR5 labeled stem cell, but really get all those cells together to do what Mother Nature does so well. And so that was our other aha moment, was understanding, instead of trying to culture and isolate cells ex vivo, because in order to do that, a keratinocyte has a totally different culture condition than a fibroblast. And so by definition, you have to separate them, grow them, and then try to recombine them. And in doing so, you're just doing a poor job at recapitulating what Mother Nature already has in place. And so out of that small skin harvest, there's millions of cells already in there, really enough to treat a large wound, even with such a small harvest. And so what we do is generate these multicellular units that have all these different cellular populations with the extracellular matrix in them that on average have a diameter of approximately 2 millimeters, which was by design because we know when you're going to put cells into a wound bed, there's not going to be an immediate vascularization. So the cells will have to survive for about 48 hours until that inosculation, inhibition, and neoangiogenesis occurs to really plug that skin back into the body. And during that time period, those cells have to live off of plasmatic imbibition, where they're just sort of living in this bath of the wound bed and exchanging nutrients. And that can only happen over 200 microns, which is 1/5 of a millimeter. And so by definition, our little multicellular units, most of those cells will be able to receive those nutrients and survive those first critical 48 hours as those cells really take within into the wound. And so when we think about chronic wounds in our first indication, diabetic foot ulcers, which Ned will talk about, we sort of consider these wounds as being biologically bankrupt. They've been open for weeks to months, something that for those of us without these conditions is hard to imagine having a wound just open for a month. You'd be like, whoa, what's going on here? But that's the case in these situations, and that's because they're exhausted. And so trying to just put a cover over these wounds and hope that these troops on the border of the war are going to refresh and be able to close is a really hard ask. And so our thought process is, let's put fresh troops, the patient's own cells taken somewhere else on the body that are healthy and unaffected, put them into the wound, and there they're able to take and expand and proliferate, as well as signal to the surrounding troops to get back involved. And so that was our sort of fundamental approach. And that's why we have about a 10-day expiration. So this was a very interesting engineering challenge for us. From the time that skin comes off the body, we have 10 days to get it back onto the patient, which in this setting is often not too challenging because the patients have an open wound. They're being seen every week by their providers anyways to get dressing changes and make sure things are going okay. So they'll get the skin harvested 1 week. They come back the following week and get the product put on. And in that sense, it was, the patient's body is going to be able to culture their own cells better than we could ever do in plastic. And so we made these multicellular units that we get back to the patient as expeditiously as possible so that they can go into the wound and graft and do their thing and really reenact natural wound healing that the body does well, but optimized for these wounds that Mother Nature's never worked on before.
As you were talking about that process, I was thinking about getting older and the fact that when I was younger, if I cut myself or hurt myself in any way, it would just heal over quickly. And now, if something happens, not only does it not heal as quickly, it's quite obvious still after about 2 years now, there's discoloration. And does the age at which the person that you're treating make a difference in that process?
Yeah, I mean, it's a great point and something that we all recognize as we get older. And it definitely does have an impact on wound healing. Obviously, if we're going to be treating a younger patient versus an older patient, you will see that inherent greater ability for wounds to heal. Interestingly, when we do a lot of profiling of cells within skin, it's hard to pick up those differences in vitro. Part of it is the tools that we have to really investigate cells at that level are, especially in a little bit more of a systems approach, not when you're just like isolating single cells, but seeing how these cells work as a group. It's hard to pick up those differences in vitro that occur with aging. On some of our assays, looking at the tissue viability, cellular viability, the ability of cells to respond to a stimulus, because during our manufacturing process, we actually trigger an upregulation in wound healing genes. That's part of our manufacturing process, is to activate the cells. We see that as long as the skin comes from a healthy area, generally we can get really good responses even from 90-year-old skin. But I think once you're putting those cells back into the wound, you're still dealing with the overall systemic environment, which can be, less blood supply to that limb, and all those different effects can impact the overall wound healing. But that is definitely an impact. And just for people with diabetes, it's well known that wounds close, 50%, less fast.
Typically, where would you take the sample from, or the skin from?
They can take the skin from anywhere. We're working in diabetic foot ulcers right now for our first indication. And a lot of that is treated by podiatrists in the United States, and they often aren't allowed to operate above the knee. So for this first indication, they're often taking the skin right below the knee on the inside of the calf, right over the muscle. But, we have preliminary evidence and other clinical use outside of that indication where, they often take it from the groin under the abdomen, but it all depends on the patient. Where's the healthiest skin that they can take easily and get closed well. So similar to any other skin graft that someone might be doing, it's kind of just unique to the patient. And we haven't seen an issue, similar to your question about aging with the skin we get in. We haven't found really any differences based on body location. So it's all about what's best for the patient and the best place to take the skin.
I know that I've seen a quote attributed to you mentioning that skin could be the first organ to fulfill the promise of regenerative medicine. I wonder if you could go into a bit more detail on that.
Yeah, thanks for bringing that up. And, when we look at the field of regenerative medicine, including tissue engineering, and think back to how did the field start and what were the approaches at the beginning, I think there was this vision of growing an organ in the lab and, really addressing the shortage of organs there are for patients that need them. And, there's even been some great work in the lab where you'll see a pretty well-developed organ that was grown in the lab. But the question is, why aren't we really seeing that clinically? And it comes down to many challenges. So if you look at solid organ regeneration, including the skin, you first need to have an organ that has the capability of regenerating itself. And then you also need to be able to not only regenerate that organ, but you'll have to insert a vascular network into it. So it needs to get a blood supply in order to get back in the body. Nick already mentioned there's often many different culture conditions you need for all the different cell types that you want to accomplish those things. And then finally, you need to have access to that organ if you're going to do an autologous approach, which until we solve the immune system, which to Nick and myself have done a lot of research in that area, it's as tricky as curing cancer. You're going to need to start with the patient's own tissue. So lining all those things up, it actually just lends advantages to doing this in skin. So as Nick said, the skin is very capable of inherently regenerating itself. There are certain situations where it can't, wound's too big, the wound's chronic, etc. But ultimately, the skin can regenerate itself. The field has pretty well mapped out how it does that, including all the different stem cell populations, their locations, their markers. We have a good sense of how it's accomplishing the regeneration. So we have a good map to start with. And there's very easy access to the patient's own skin. So unless you have a 99% burn on your body, they're probably going to be able to find skin in order to treat it with a regenerated product. And another unique feature is we don't necessarily need to integrate a vascular supply into what we engineer because you can put it back on the wound and it's able to gain its vascular supply by engrafting. So taking all of those features into account, it just sets skin up to be able to lead the field, to regenerate itself, to create products and therapies that are able to leverage all these unique attributes in order to take the field into a new era. So by no means do we think skin will be the only one and that no other organ will be able to be regenerated or engineered the way we've envisioned. But from what we see, skin may be able to be the first and help lead the field into new approaches. And I think the other thing we're starting to see is groups take different approaches to the solid organ challenges that I just mentioned. So maybe instead of what we envisioned at the beginning, there needs to be other approaches. One example is we've recently seen engineered pig organs being transplanted into humans. So instead of trying to create a whole new organ in the lab, they're taking a supply of organs, genetically engineering them to go back to humans. But that's just a first step because those organs you still need to take immunosuppression to receive those organs. So it's starting to address the shortage of organs if this path works, but it's not going to necessarily get rid of the need for immunosuppression. So we start to see different approaches coming up with the other solid organs. And then I think if you look at our approach to skin regeneration, it doesn't look exactly like how tissue engineering was envisioned in the beginning, where we're optimizing the skin in the lab, in the manufacturing process, and then getting it back to the body for the regeneration to occur instead of expanding it in the lab. So we think there's a big opportunity for skin to lead the way, and there's also just a massive need for it. When you look at chronic wounds, there's approximately 8 to 10 million in the US every single year. If you look at diabetic foot ulcers specifically, which we're attacking with our first indication, there's 1.6 to 2 million patients affected every single year with a diabetic foot ulcer. You need to be able to scale a technology into a disease that common. Essentially, every 20 seconds, a patient in the US gets a diabetic foot ulcer. Every 3.5 minutes, someone gets an amputation that was started with a diabetic foot ulcer. So, as opposed to going after a rare disease where very few people need it, and you might be able to have a highly complicated lab incubation time for an organ, we're talking about a very common disease affecting millions of patients. And our approach will be able to scale up to address that need because of the way we've designed the product. So we see a lot of reasons why our approach could be successful for regenerative medicine in a way we haven't seen hit the clinic before. But just skin as a whole is a huge unmet need that needs to be addressed before a lot of other things.
Are there other companies working on similar things with the skin or with other organs?
I think from our approach, nothing that we've seen exactly this way. I mean, there's been companies that have taken allogeneic cells, taken allogeneic keratinocytes, allogeneic fibroblasts, cultured them on a collagen scaffold, and then those dressings, they're were approved through the FDA as a dressing, so not as a drug. Again, primarily just providing a coverage, not really integrating. So there's been approaches of trying to involve cells in wound treatments. There's cadaveric skin that's either been decellularized, so it's just the extracellular matrix, or there's cadaveric skin that's been cryopreserved and frozen, which still retains its cells, or even fresh cadaveric skin, which still has the cells, but as Ned mentioned, it's allogeneic. So again, they're just providing a coverage until the body rejects them and boots them off. But a really autologous approach that's engineered, other than an autologous approach of just surgery, where you're cutting out from one area and just really laterally transferring that tissue, we haven't seen a similar approach to ours. And then, as Ned mentioned, once you get to more solid organs, especially within the body, that approach is even more limited just by constraints of getting that source tissue to begin with.
And I think, on a different side of the fence with rare disease, genetic disease, we have seen successes in skin. So Abeona and Crystal Biotech both have regenerative products for epidermolysis bullosa, so a genetic disease where they're missing or they have a mutated collagen 7A gene, doesn't allow the skin to anchor to the dermis. And they have taken different approaches to replace that mutated gene. Abeona essentially takes the patient's skin, recultures the keratinocytes with the gene being replaced and puts it back to the patient. Crystal Biotech essentially has a cream with the gene in it that would then go on to replace the gene in vivo on the patient. So even in the rare disease space with regenerative medicine, outside of the CAR T space and cancer and autoimmune disease, we're still seeing skin kind of at the leading edge of what's being done. And, I think it's exciting to see those companies getting their approvals and starting the commercialization process.
We've been talking primarily about wounds, but does this have implications or potential uses with other skin conditions, whether that be genetic conditions, skin cancer, those kind of things?
Yeah, I think when you look at other conditions, our technology, the product itself was designed for wounds, but as you're talking about these other areas, our first indication is diabetic foot ulcers, but we plan to expand that into other wound types. So even with skin cancer, ultimately this product could be needed to reconstruct a wound after the cancer is removed. But we're also looking at pressure injuries or, what used to be called bed sores as the next indication, and then moving up the ladder into acute wounds, traumatic wounds. One area where there's a lot of need is when the wounds are deep, they're contoured, and they're exposed structures like bones, tendons, things you can't put a normal skin graft on, and you need to build the tissue back up. We have preliminary clinical evidence that skin TE may have value in that scenario. So those are areas we're looking to take skin TE. I think the base technology has a lot of potential in regenerative medicine as a whole, but we're staying laser focused on skin, on wounds, for the foreseeable future, but could definitely take this outside of skin and wounds if there was motivation to do so.
Are there any complications or challenges of working with the skin and wounds? I mean, there must be a point at which a wound is too big to be able to handle, for example.
Yeah, our product, if you take more of the skin harvest, then you can treat larger wounds. And as Ned mentioned, we have preliminary data treating wounds that were even thousands of square centimeters. So there's the ability for the product has about an expansion, at least in our preclinical data and early clinical data, about 1 to 23, meaning if you take harvest about a centimeter squared of skin, you can treat about 23 centimeters squared of skin from that tissue harvest roughly. And that's partly because, again, there's millions of cells in that small area of skin that we're really optimizing and activating for that wound healing. It's much more potent, if you will, than if you were just doing a little skin transfer of that harvested skin. As far as challenges you really start to deal with the challenges of just managing that patient. You can imagine if you have a very large wound where your entire arm is involved, you're going to be in the hospital for some time. And it's really a race of getting that wound closed before things like desiccation, scarring, and infection set in. Diabetic foot ulcers present a unique challenge. They're on the foot by definition, and you're often having to walk on your foot. So it's very hard to tell a patient, we're going to put this, cellular biologic onto the bottom of your foot and try not to walk around for, 2 weeks or so, which would be ideal. And in many other traumatic situations where patients will be admitted to a hospital because they have a lower extremity traumatic wound, it's not uncommon if they get skin grafted that they're on bed rest, for, 5+ days. But with how common DFUs are, affecting well over 1 million Americans, and that these individuals are trying to go about their everyday lives, we now have to try and integrate this sort of cellular complex autologous biologic in with the patient's everyday life, and really making sure that the dressings that are put on can protect that wound, and that we help those patients get around and navigate their life while trying to maximize their wound healing potential.
Yeah, I think if you look at wounds, all the advantages I described with skin regeneration, easy access to the skin, the wound can provide the vascular network for the product that's engrafting. Also comes with it one of the most unique challenges of any regenerative product you can think of, which is essentially what Nick's describing, the wound's in communication with the outside world. So you're going to need to put dressings on it. You're going to need to manage that interaction with the world, whether that's pressure, traumatic injuries, moisture, desiccation, creates the follow-up challenge with dealing with wounds and skin that I think can't be underestimated. You could have a perfect product in the lab, but if you get into the clinic and you get into the real world with patients and wounds, you're dealing with a whole different animal that you really can't model out with any preclinical studies until you get to your clinical trials.
And are there any hurdles like regulatory, or you mentioned the number of people that have these issues in terms of numbers of, what was it, every few seconds somebody is diagnosed, in terms of being able to deal with all of those right across the country? Is that going to be a big hurdle?
Obviously, there's a large amount of wounds, and I think the regulatory wise, we're pursuing the BLA pathway, and that has been going quite well to date. We got breakthrough therapy in March of this year granted by the FDA. We previously had a regenerative medicine advanced therapy RMAT designation granted by the FDA. And so it's been a very productive and actually quite a fun process, working with them, trying to get this new class of autologous multicellular biologic into, the hands of patients, which, we will be first in class. And so those hurdles are obviously always challenging. It's a rigorous process to get that approval. That's been going quite nicely. And as Ned mentioned, we'll be submitting for our BLA hopefully around April of 2026 and get approved by end of 2026, early 2027. So that hurdle is there, going well. But to the second part about the number of patients, obviously there's unlimited amount of patients essentially to treat out there. And the challenge is there, not only with manufacturing, our process is unique because every single patient is their own lot. So this isn't a traditional scale up process where instead of making a liter of product, you're making 10 liters of product. This is really scaling out where you're trying to build multiple parallel manufacturing pathways because you're now just making multiple lots at the same time. And that chain of custody, ensuring that that patient's harvest that comes in is going back out to that same patient, because obviously if products would get crossed, now you're going to have a rejection reaction and the product's not going to do any good. And now you've, impacted two patients is a critical aspect of our manufacturing that we have to pay very close attention to and spend a lot of infrastructure in building around. Our capabilities here in Salt Lake City, we have over 60,000 square feet of facility space where we have modular clean rooms that we built within it. We have quite a bit of capacity in order to meet those demands. Obviously, to get to multiple millions will take even more scale out, but we feel pretty comfortable with our capabilities so far to handle, launch and beyond. And then the other part is really getting providers also properly trained in the process, because although it's quite simple, taking a little skin harvest, kind of like getting a mole removed, something that can be done in the office, generally fits. We've found in our clinical trials, it fits well in their clinical practice, but it's a little bit of a new process, new procedure. And then again, having, everyone, including the patient, appreciate these are living cells that are going back into your wound bed. And as Ned mentioned, you really have to protect them from the outside world, meaning not smashing them and letting them really take and engraft is a part of that training process as people get used to this product to make sure they follow so that the wound has the best chance in closing.
And you've mentioned the fact that this is clearly something that's autologous, but do you ever envisage that there's a potential for this to become something that's an allogeneic treatment?
It's something we've looked into and obviously thought about it. Having something off the shelf would be fantastic, and that would be the holy grail. And as Ned mentioned, until you can really address the issues of immunity, which is as complicated as cancer at that point, it's going to be a long way off. And thankfully, we already have a pretty good solution for it. Where we can see some off the shelf capabilities would be, again, as we mentioned, a small amount of tissue can treat a very large area. Oftentimes we have more than enough skin TE, especially for these smaller wounds, where maybe we can freeze some of that and bank that here. And so if that patient develops another DFU, which, anywhere from 40 to 60% of these patients are going to be developing another DFU in their lifetime, they very frequently recur, that the provider, instead of having to take another tissue harvest, could call us and we can thaw out the skin TE and send them another dose at a later date. Additionally, I think there's some possibility of us incorporating some allogeneic cells, maybe some more immune privileged cells, such as mesenchymal stem cells that would be allogeneic, that we could use to augment our autologous product. So especially when we're dealing with these much larger wounds, could we incorporate some of those allogeneic off-the-shelf cells in order to be able to treat even larger wounds with a small tissue harvest?
And could you tell me a bit more about some of the clinical trials that you've been involved with?
Yeah, so we've run 7 clinical trials to date. 4 of those have been in diabetic foot ulcers. As Ned mentioned, we're very excited by just a little under 2 weeks ago, our last visit occurred in our phase 3 pivotal trial in Wagner 1 diabetic foot ulcers. And so in those trials, we were comparing patients being treated with our product with standard of care versus standard of care alone. And so went through a pretty rigorous process where patients would come into the trial, they'd get 2 weeks of just rigorous standard of care. And then if their wounds didn't respond if they started to close, great. They would just continue on with standard of care, wouldn't go into the trial because they're already on a great trajectory of healing. But we really focused on those wounds that didn't get smaller. And then those patients would be randomized to either standard of care plus skin TE or standard of care alone. And then they would get a treatment, and then we would follow them for 6 months, with the primary endpoint being wound closure within the first 3 months. And this is a pretty standard trial design that's been done in wound care in quite some time. And our phase 2 study was a very similar design, a little bit smaller overall patient population, a little bit shorter follow-up period. But in that study, skin TE treated wounds closed 74% of the time versus 34% with standard of care. So, roughly, double there with skin TE effect in these very difficult to treat wounds.
And could you tell me a bit more about the rest of your pipeline and other things that you're working on?
We're staying laser focused on developing skin TE into other indications. So essentially, pipeline in a single product. And with the preliminary evidence we have in other wound types, we're planning to build out within the chronic wound space first. So as I said, pressure injuries, also formerly known as bed sores, and venous leg ulcers both have a huge unmet need and are in essentially the same sort of channel space providers that are treating the diabetic foot ulcers. So works well into expanding the product into those indications commercially. And then beyond that, we're really focused on getting into acute and traumatic wounds. So essentially acute skin defects that are just too large or too deep or have exposed structures unable to be grafted through traditional means. And, the product could go all the way into burn. That's a little bit further down our list in our pipeline. But our plan is to get the product back into every wound type that, it could provide value in. And we think we have a good sense in pressure injuries, venous leg ulcers, and acute skin defects could be a great path forward. And then there are all sorts of other wound types that fall in between the cracks of definitions of all those indications that we'll have to kind of evaluate on a one-by-one basis, or see if we can get some creative trial designs to group them together and get a broader indication on our label that could address everything. So that's where we're focused for now. That in itself would be sort of a career worth of work for us. But, let's say we get through all that. We also get the cryopreserved version out onto the market and get that on the table. There was always sort of visions of taking this beyond skin, but in the development and maturation of the company, we've realized we've got something pretty special in skin TE, and to just want to stay focused on wound healing and skin regeneration before we start getting distracted by other applications of the technology.
Where do you see the field going? I mean, not just necessarily for your own product, but how do you see the field of skin regeneration?
I think we're seeing the field actually hit a big inflection point for evolution. Wound healing as a whole, wound care. We saw it first with the FDA having products come down this BLA pathway that we're in. And we think other advanced products are going to need to go down this pathway. So transforming this field away from iterative innovation into very different, very innovative products, biologics, cell therapies, et cetera. We see it going this way. We see the field consolidating. So right now, there's about 220 skin substitute products on the market in the United States. Medicare is pushing forward a lot of changes that may or may not go live in January that will force a consolidation in the market. But we think even with what's already happened, a few of those products will rise to the top, and a lot of the iterative products will probably go by the wayside. And we'll see wound care start to have less and less of kind of the menu of options that we see today, where you could pick one of 40 different amnions you want to use to put on a wound. It may bubble up to leaders in the field, kind of leading each category of wound dressing, where, we hope to come in with an autologous multicellular therapy to lead that definition. We'll be first in class there. And as the field evolves in this way, we think it'll also start to bring the field to mimic what you see in other fields of medicine. So wound care right now isn't actually a recognized specialty within the American Board of Medical Education. You can't go do a wound fellowship that's recognized by any specific society or body. There are many out there, but they don't fall under the ABME right now. And we see that evolving as well. We think this is a huge unmet need, and it's not just a lack of innovation in products, it's the whole ecosphere. So we foresee in the future providers pursuing very specific wound healing training that needs to encompass not just surgery, not just managing the wound, not just internal medicine and managing the patient, but the whole thing. So it may be the very beginning of all these changes, but there's sort of undeniable proof that the field is starting to move away from sort of the iterative product menu that exists today and more into a field of true medicine.
Some really interesting science there, and let's hope that diabetic foot ulcers and other wounds can soon be taken care of extremely quickly and efficiently. It's definitely a huge field, that's for sure. So that's it for this week's podcast, and indeed for this year. If you do want to get in touch with guest opportunities, or if you want to advertise, please do feel free to contact us at jim@deeptechdigest.com. I did think of taking a couple of weeks off over the holiday season with the podcast. Definitely Boxing Day here in the UK, there will not be one, but as we have so many interviews still to get through, we will be back on January the 2nd with episode 41. So, until next time, wherever in the world you are, I hope you have a great holiday season if you are celebrating. Thanks for listening, and I hope you'll join us again next year for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.